Welcome back. In Unit 5, we'll talk about sensation and perception. In 5.1, which is this video, we'll talk about sensation. And in 5.2, we'll talk about perception. So how is sensation different from perception? Now, in the colloquial expressions, we use sensation and perception interchangeably. In fact, we mean perception when we say sensation. I have these sensations. When you say that, you're actually talking about perception, because when you say I have these sensations, you're referring to conscious experience. And conscious experience is not a part of sensation. Conscious experience is a part of perception. So, just to make it clear, sensation is the capture of information by your sense organs. It happens at the level of sense organs. That's why it's sensation, sense organs. Perception, on the other hand, happens at the level of the brain. It is interpretation of the captured information by your sense organs. So if sense organs are capturing external information, perception is the brain interpreting the information that is sensed by your sense organs. So this is at the conscious level. This is not at the conscious level. So sensation is mere capture, and perception is interpretation of the captured information. Also keep in mind that not everything that is captured by your sense organs is interpreted by your brain through perception, but only a part of what is captured is being perceived or interpreted. And as we discussed in the big picture, what helps us from going from a lot of information to little information is the attention. The outcomes for this video, which is visual sensation, uh, are listed here. And we are only talking about visual sensation because this is an important area that is searched more than other sensations, but also because visual sensation is key in solving the puzzle. Remember, the puzzle is to understand how our brain comprehends the written text. And when you have written text, you need to be able to capture the printed information. Can you close your eyes and read what's on the screen? No, you didn't. You need your eyes to capture the printed letters so you can interpret it. So that's why we only pay attention to the visual sensation, not sensation in other senses. So in this particular unit, we'll talk about the structures involved, the basic structures involved, some important parts of the brain, and most importantly, the eyes. Then we'll talk about the process of visual sensation, which includes a construct called compression. We'll talk about that. And also we'll talk about how our eyes capture light, which is basically why we see things, and which is basically why we are able to actually read what we see on a text or in a text. So the structures involved in visual sensation include the eyes and the brain. So we'll talk about the eyes and then a little bit about the structures of the brain involved in visual sensation. Now, this is the structure of the eye. Now, if you can see, this is more like a small vessel or a pot or a socket filled with fluid. And this particular socket is covered by this structure called the lens. And the light passes through this lens, and because of refraction, an image of whatever you're looking at will fall on your inner layer of your eyeball. So the basic structures include lens then, and the lens is covered by this muscular structure. It's a circular muscle It's a circular muscle, just like the shutter of your camera. And this muscle can constrict or dilate, changing the opening that's left here, or the size of the opening, as a result of the movement of this circular muscle. The circular muscle itself is called the iris, and the opening that is left by the circular muscle is called the pupil. So when there is more light, then the iris muscle will constrict, reducing this opening, and less amount of light passes through the pupil, passes through the pupil, through the lens. If there is less light, then your iris retracts and creating a larger opening, so the light will enter through this and passes through the lens. Now, coming to and this is the protective layer of cornea, transparent layer. So light first passes through the cornea, then passes through the pupil, then the lens, and an image is formed on this innermost layer of the eye. And this innermost layer, which is in yellow color here, is the retina. So we'll talk about the structure of the retina in the next slide, but just want to give you a bigger picture here, in the sense that there is a structure here called fovea, and this is a part where there are more photoreceptors of a specific kind, um, and we'll talk about what these are in the next slide. And this is the most it's like the center of the eye, in the sense that if you're paying attention to something, and it's called visual area of visual acuity. If you're paying more attention to something, you'll know more details of an object if the image is formed right here. Now, if an image of an object that you're looking at, when it passes through the lens, does not fall exactly on retina, it may fall in front or behind retina, based because of some an distortion in the structure or the lens of the eye, that results in defective eyesight. And through glasses, we can fix it, so that if the image is formed in front, now the glasses will fix so that the image actually is formed on the retina. And so also if it is formed beyond the retina, the glasses again will fix it so that the image is formed on the retina. So for the eyes to capture whatever you're looking at, information about it, the image of it should and must need to fall on the retina. Now we look at the structure of the retina in the next slide. The retina has three layers of cells. First layer is here, the second layer is here, and this is the third layer of cells. The first layer of cells is comprised of photoreceptors. These are photoreceptors, they are specialized neurons that are sensitive to light. And when light falls on these photoreceptors, they convert this light into neural impulses. And this process itself is sensation. So the innermost layer is the layer of photoreceptors. There are two kinds of photoreceptors, rods and cones. Rods are meant for dim light vision. In other words, they help us identify the object as opposed to give more details about the color. Cone, on the other hand, cones are for color vision. They give more detailed information about the object. In fact, there are more rods than there are cones. Again, rods for dim light vision and cones for color vision or daylight vision. Now, the second layer of cells is the bipolar layer cells, right here, the bipolar layer cells. Now, these bipolar layer cells are mere connections between the photoreceptors and the next layer of cells. The next layer of cells are called the ganglion cells. Ganglion cells. So one more time, photoreceptors, bipolar cells, and ganglion cells. So when light enters through the pupil, the lens, right, it goes through the bipolar cell layer, the ganglion cell layer, the bipolar cell layer, and eventually it reaches the photoreceptors. And because the bipolar layers and the ganglion cell layer are not sensitive to light, nothing happens there. Once the light reaches the photoreceptors, then for the photoreceptors actually create neural impulses, the action potential. These action potential generated here in the photoreceptors will be transferred from photoreceptors to the bipolar cells, from bipolar cells to the ganglion cells. And the axons of the ganglionic cells, they come out of each eyeball as a bundle of axons forming a nerve called the optic nerve. So the axons of the ganglion cell layers go through a specific part of the eyeball where there are no photoreceptors. This is the part called the blind spot or the optic disc. This is a part where there are no photoreceptors. This is a part where the ganglion cell layers axons form a bundle and come out of each eye as optic nerves. So the information that is captured by the eye from each eye, right? It goes from the eyes to thalamus, and from the thalamus, remember the thalamus, a subcortical structure. From the thalamus, the information then is sent to the occipital lobe, which is the visual perception center. There's a visual cortex in the occipital lobe. That's where the information is sent to. And information from both the eyes goes through this. And if you remember, the information, the fibers are these are optic nerve fibers coming from each eye, right? And they crisscross in a way that they form a structure here called optic chiasm, if you remember from last um, video. So they form a structure called optic chiasm, right? And this also should remind you about how the information is the system follows contralaterality, but not at the level of the eye, but at the level of the visual field. In other words, the information in the left visual field is eventually sent only to the right occipital cortex, and the information in the right visual field is sent to the opposite occipital cortex. So contralaterality in vision is at the level of the visual field. Now that we talked about the basics of the structures involved in visual sensation, which includes the eye and the visual pathway and the brain, now let's try to understand the process of visual sensation. In other words, not how, but once the information is captured by the retina, what happens to this information? Then we'll talk about how actually eyes capture information. If you remember, the retina has three layers of cells: the rods and cones, the bipolar cells, and the ganglion cell layer, right? So the information captured by the rods and cones here gets transferred to bipolar cells and then to ganglionic cells. And from there, it goes out through the optic nerve fibers. Now, an important point to notice here is that first of all, there are more rods, as you can see here, than there are cones. Even in this single layer, at this specific point, right, there are more rods than cones. And throughout the retinal layer of the eye, you'll see more of rods and less of cones. In fact, there are 120 million rods in each eye and only 7 million cones. In total, there are 127 million photoreceptors. So your photoreceptors, 127 million photoreceptors, 27 million photoreceptors, um, um, they capture the information from the light, from the outside. But the issue here is that there are only 1 million ganglionic cells. As you can see, the number decreases as you go from photoreceptors to bipolar cells to ganglionic cell layer. So just think about it. Think of these photoreceptors as the pixels in your camera. So these retinal cells, rods and cones, are capturing, this is just an analogy again, capturing 127 million pixels of information. But then once the information is sent to the ganglionic cell layer, the ganglionic cell layer has a lesser resolution in the sense that there are only 1 million um, cells. So what should happen here in terms of 127 million pixels of information should be converted into 1 million pixels of information. In other words, information must be compressed somehow. And in fact, it is compressed. It's called compression. Information from captured by your photoreceptors is compressed so that it can fit the fewer ganglionic cells that can capture the information from the rods and cones, and that is sent again um, to the brain through optic nerve. And this process is called compression. And when we say compression, what we are basically saying is that, now again, keep in mind, this is the information is sensation ends here, right? In other words, the capture of information by your photoreceptors is sensation. Now the information is sent to the brain so it can perceive, interpret the information. But before the information is sent to the brain, right, it is compressed right here at the level of the eye. So you can think of it, this compression is more as summarization. Information is compressed in a way that the most important information is definitely captured and sent to the brain. And the important details of information include the edges, the contours, the shades, information sufficient for you to actually detect what an object is, because that's what perception basically is, which is interpreting what the captured information is. Just identification. So this process is called compression. So we kind of looked at like we're going backwards, right? If you think about it, we said that the retinal cells, in fact, the photoreceptors capture light, and that is sensation, correct? And then we talked about what happens to this captured information, right? Then we talked about where the information goes to the brain, right? Now we'll talk about how the information is captured by your retinal cells. That brings us to the next outcome, how eyes capture information. Again, keep in mind, we are only talking about what happens before compression. It's how the eyes actually capture information from the external world. So that would be the last outcome, and that's what we'll talk about uh, in the next few slides. Now, here is the question. Now, stop whatever you're doing and try to do this task. Now, try to look around, right? Or try to see an action. Maybe look at your friends who are talking. Now, think about it. You're experiencing this event that's happening in front of you continuously, right? It doesn't feel like there is gap. There is no. It's like a continuous process without any gaps and delays. Correct. Now here's the question: how do our eyes capture this information? One analogy is it captured like a video where everything is captured continuously, or is it captured intermittently? Just think about it. Pause here and think. Is it a continuous capture of information, or is it's intermittent capture of information? Well, if you said intermittent capture of information, you're absolutely right. Our eyes don't capture light or information continuously. In fact, your eyes your eyes go through a jerky moment, right? A quick jerky moment of the eye. It's called saccade. And this is what you can do. You can call your friend or your family member sitting next to you and ask them to stay somewhere and try to see their eyes. Even when they're not reading, the eyes will make jerky movements, and these movements are called saccades. And during saccade, your eyes can't capture information, right? It's only between saccades, between moments, that's when it captures information. And that is called fixation. So, so just to give you an idea, right? Oh, if you're say reading a sentence like this, right? After a long day at work, Mr. Blah 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 enjoyed, right? So what happens is your eyes fixate on the words, okay? And during this fixation is when your eyes capture information. Then your eyes move from one word to another, and this movement is called a saccade. So there is a fixation and a saccade, fixation and a saccade, fixation and a saccade, and your eyes are blind during saccades. They only capture information during fixations. And you go through fixation saccade cycles. You go through fixation saccade cycles, which roughly lasts for, let's call it 250 milliseconds for our convenience, for around 250 milliseconds. Of this 250 milliseconds, 200 milliseconds are spent on fixation, and 50 milliseconds are spent on saccade. In other words, you go through 200 milliseconds of fixation, your eyes don't move. Then for 50 milliseconds, they move. Again, for 200 milliseconds, they don't move. And for 50 milliseconds, they move, and so on and so forth. So each SF cycle lasts, let's say, 250 milliseconds, right? 200 milliseconds of fixation and 50 milliseconds of saccade. And your eyes go through this moment several times. Now think about it. In a second, how many SF cycles do you think your eyes go through? That's right. Around 4, right? 3 to 4. 250 milliseconds, 250 milliseconds. So how many 250 milliseconds you find in 1 second? Millisecond is 1,000 seconds, right? Sorry, 1,000 milliseconds will make 1 second. So in 1 second, your eyes will have roughly 3 to 4 SF cycles, 3 saccades and 3 fixations. Okay, so I've tried to create this visual representation of, say, one fixation, one saccade, followed by a fixation cycle. So a box here represents a fixation, a box here represents a fixation, right? And the length of this bar or a box represents the time, right? So there is one fixation for 200 milliseconds, there is another fixation for 200 milliseconds. And in between fixations, your eyes move. So I represented this as this empty place called a saccade, quick eye movement, so that you go from one fixation to another fixation. You go from one word to another word. Okay. So, and we said that this is for 200 milliseconds and 250 milliseconds. This is for 50 milliseconds, for a total of 250 milliseconds. And only during fixation does your eyes actually capture any information from the external world. During saccade, it actually is blind to the external information. Now, this is what I want you to do. Pause the video here, then find a mirror and try to see if you can see your own saccade. What is it? What do I mean by that? Well, try to look at yourself, look at your eyes, right, in the mirror, and try to look away and come back and try to look at your eyes again. And try to see if you can capture your eye movements while you're looking away and looking back at your eyes. The question is, can you see your saccades? I'm going to pause it and ask you to actually see if you can find a mirror and see. Don't use the camera phone. I know it's tempting to do that. What did you see? Can you actually see your saccade? Well, you're absolutely right. You cannot see a saccade because you cannot see a saccade because during a saccade, your eyes don't capture any information. It's completely blind to the external world. It's completely blind, so there is no way you can see the saccade. But if you use your camera phone or a camera of your phone, right, then it's likely that you may see your eyes moving. And the reason for that is not because you can see your saccades, it's because of the buffer of the image of your moving eyes that's captured by the camera. So you can't see your own saccade. And this blindness during a saccade is called inattentional blindness. Now here is another interesting thing. We said that a fixation lasts for 200 milliseconds, and a second, and followed by a saccade which lasts for 50 milliseconds, followed by another fixation, and the process continues. Right? That's why it's a cycle, FS cycle, the fixation saccade cycle. Now, here is an interesting thing. We said that you can't see your own saccade because your eyes are blind at that point, right? So the 50 milliseconds, your eyes are blind, right? And we said that your eyes only capture information during fixation, which is for 200 milliseconds, right? But here is another interesting thing. Your eyes don't capture information for the entire 200 milliseconds. They only capture information during the initial 50 milliseconds. And for the remaining 150 milliseconds of the fixation, your eyes are blind again. What? Yes. So, so if you take one fixation and saccade cycle, which is 250 milliseconds, out of 250 milliseconds, 50 milliseconds, you're blind, right? Plus for 150 more milliseconds, you're again blind, which is like 200 milliseconds. The only time your eyes actually capture any information is during the initial 50 milliseconds of the 200 milliseconds. So if you take one cycle, which has 250 milliseconds, you're only capturing information during 50 milliseconds, and for the remaining 200 milliseconds, you are blind. So in one SF cycle, for how long is your system blind? For 80% of the time. 200 for 200 milliseconds of 250 milliseconds, you're blind. That's scary, right? Or if you take 1 second, right, which has 3 SF cycles, right? So for 600 milliseconds, like 3 times 200 is 600, right? For 600 milliseconds, you're blind, and the remaining 400 milliseconds is is it 150 milliseconds is when your eyes are actually capturing information. So around 70 to 80% of the time, you are blind, and during only the 20% of the time, your eyes are actually capturing information. Um, so we said that your eyes are blind most of the times. 75 to 80. Again, it depends on the length of the fixation. It's not exactly 200, it's 200 to 250 milliseconds, and if it's 350, 250, then the number of cycles you have will change from 3 to 4. That's what's roughly 75 to 80 or 70 to 80%. So 70 to 80% of the time you are blind, and for the remaining 20 to 30% of the time is the only time your eyes are actually capturing anything. It is scary, right? And you can generalize that for the entire day with your eyes open. If you're awake for 12 hours, what percentage of the time you think your eyes are actually capturing time information? Well, only 20 to 30% of the time. For the remaining 70 to 80% of the time, you are blind. Anyways, here is an interesting question. If you take one SF cycle, all right, and if you're only capturing information during the initial 50 milliseconds of the fixation, what happens during this 200 milliseconds when our eyes are blind? In other words, during the 150 milliseconds of fixation plus the 50 milliseconds of saccade. What do you think is happening? Why don't we feel the gaps? Think about it. Pause here for a second. I seriously mean it. Pause here for a second and come back. Okay, I hope you paused and thought about it. Well, the reason why we don't feel the gap is because whatever is captured during the initial 50 milliseconds, right, that is being perceived by the brain for the remaining 200 milliseconds. Does it make sense? So your brain is capturing, your eyes are capturing information, is capturing information during the 50 milliseconds. When the remaining of the SF cycle for 200 milliseconds, your brain is actually perceiving, interpreting, making sense of what is being captured, so that your brain is busy processing the captured information. Then it will capture information again. Again, the brain is busy sensing the perceiving the captured information. That's why you don't feel that delay. You don't feel that gap. So, so what happens when the eyes are blind? Well, you don't realize they are blind because your brain is actually kicking in and actually it is perceiving what is being captured. Well, perception then. So, sensation is mere capture of information by your eyes, which your eyes do it by intermittently capturing information during fixation scheme, only during the 50 milliseconds of the fixation. For the remaining time, you are perceiving. Now, the next question is, what is perception then? How does your brain perceive the captured information? That we'll talk about in the next video, 5.2 perception. Well, I hope you liked it. Um, have fun learning. Thank you.